Close my eyes, but it's too late. You slip through my fingers. You slip through my fingers. You slip through my fingers, baby. You slip through my fingers. You're like water in my hand. Water in my hand. You're like water in my hand. Water in my hand. You're like water in my hand. Water in my hand. You're like water. Water, water, water in my hand. Blossom my branches for all to see. And cherry my hair, set me free. Sun me over, breathe through my leaves. I'll baby choose my fruit. Of my lips, I send my soul winging. Oh, send me to him and start me over. Breathe and tell me, oh, fill my cup and drink from me. Patience I She fell to the ground, her soul flew over me. Eyes wide, no sound allowed, but the wall screamed right through me. As she sank into bed, suffocation, the life she led. No sound I made out loud, but the walls came tumbling down. Could you leave me?
My name is Ann Armstrong. I'm the manager of OMSI's Coastal Discovery Center at Camp Gray, and tonight I'll be your host as we learn a little bit more about the brain and time. OMSI is committed to sparking curiosity and igniting imaginations. To help everyone at home, we've crafted and curated all sorts of engaging and science activities and experiments to help inspire you and experience the wonder of science from wherever you are. Visit omsi.edu for more information and resources. And if you do any of these science activities, please send us a video or some photos so we can see how great a scientist you are. I also hope you enjoyed tonight's pre-pub trivia and music by local bands Wildbird and Redbird. They are currently scheduled to perform live in OMSI's Planetarium as part of our Kindle concert series on October 4th. You can find more information at our website. Putting on all of these live shows takes a lot of work, and we have amazing partners to help us make this happen. A big shout out and thanks to Celestream for providing the live streaming services to tonight's Science Club. We really appreciate their support. We are excited to welcome you back to the museum and experience Body Worlds and the life cycle and the tour, the USS Blueback. So advance tickets are required. The health and safety of OMSI's guests and staff is the utmost of our priority, and we want you to feel comfortable and safe while at OMSI. To meet state guidelines and help limit the spread of COVID-19, OMSI has implemented some changes throughout the museum. Visit omsi.edu for more information. OMSI is hosting our second virtual OMSI After Dark celebrating Oregon beers. Tickets are $40 and include 10 beers and 2 tasting glasses, and access to the virtual event, which will be held on Friday, September 18th from 7 to 9:30. You can pick up your beer box at OMSI or have it mailed to you for $15, Oregon residents only. The virtual program will include science demos, brewery experts, live ice carving, and more. Visit omsi.edu/brewfest for more information. And guess what? There's something you can do to support your local community. We have some outstanding food and beverage partners, and I would encourage you to put the pub back in Science Pub experience. So go ahead and order some delicious food and tasty beverages from one of our partners from around the state. Tonight's event will be very similar to our regular Science Pub program. For those of you who don't know what a regular Science Pub looks like, we're going to begin with a brain-themed trivia that is a warm-up for tonight's talk. So grab a pen and paper so that you can participate. After that, we'll have our lecture by Dr. Mazzucato. For the Q&A, you can submit your questions at any point during the lecture via the comments on our live feed. We will collect them all, and after the lecture, I will ask your questions to our guest speaker. If you enjoyed tonight's lecture, please consider making a donation or purchasing a Science Pub pint glass. The link is in the comment section. But don't worry, there's no pressure to donate. Our mission here at OMSI is to inspire curiosity by creating engaging science learning experiences for people of all ages and backgrounds. So sit back and relax and really just get ready for a good lecture right after the trivia. All right, this week for trivia, we have OMSI's gift officer from our development team, joining us to play along with you at home. Please give a warm welcome to Courtney Cooper.
Hi, everyone, and hi, Anne. Thanks for having me on. I'm excited to play trivia and learn more with all of you.
Fantastic. Thanks for coming out and great to see you. Is there anything specific you are most excited about from tonight's show?
Oh, I'm excited for this whole thing. I took a class in college about time and the physics of it. So learning more about time and how it works in the brain is something I'm really looking forward to.
Awesome. It's always great to connect new things with things you learned in the past to reinforce all that.
Yeah.
All right, let's get started with our trivia game. So grab a pen or paper and you can play against Courtney or play against your family. You can make it a game. You can do it for bragging rights. You can do it for who's doing the next dishes. Courtney, if you get all 10, I'll figure out how to give you a cookie. I don't know how that'll work out, but it'll happen. But you got to get all 10. There are 10 multiple choice questions. No pressure, of course.
All right.
I'll read the question out loud, give you time to guess, and then I will reveal the answer before moving on to the next one. Okay, you ready?
I'm ready.
All right, here we go. Are there A, more neurons in the brain, or B, more stars in the Milky Way?
Ooh, that's a tough one. Well, I know that there are hundreds of billions of stars at least in the Milky Way, and lots of neurons in the brain for sure too, but I think I'm gonna go for small things or big things. This is the toss-up here. I think I'm gonna go for more stars in the Milky Way.
More stars in the Milky Way. No, there are more neurons in the brain.
Wow, that's like more particles of sand on a beach as opposed to in a galaxy too.
People are counting a lot of stuff.
I bet some of our viewers got that one right though.
All right, number 2. Which device acquires more bits of information per second? Your phone while you video chat, your laptop while you are streaming Netflix, or your brain?
Well, I think it's the brain on that one, because with the brain, you're taking in so much information about not just visual and audio, but all kinds of smells and other things too. So I'm going to go with your brain.
Let's see. Yes, you picked up on tonight's theme. All right. Yes, your brain acquires more bits of information per second than any of those other devices. All right, number 3. Which part of the brain did Leonard Shelby lose in the movie Memento? The prefrontal, the cerebellum, or the hippocampus? Did you see this movie?
I did. Actually, in the time class that I took in college, we watched this movie. And I remember that in that movie, he has a hard time organizing his memories. And I also know that the hippocampus is the part of the brain that deals with memory. So I'm going to say that the answer is C, the hippocampus.
Yeah. What I remember about this movie was he couldn't make new memories. And so he couldn't remember things that happened yesterday or a week ago because he couldn't make new memories.
Yeah. And it was like you were watching the movie backwards, because it would show you one thing and then it would show you what happened before that.
Yeah. All right. Let's see. Hippocampus, you are right. Okay. Number 4. What fun stuff can neuroscientists do with optogenetics? Can they genetically engineer neurons to be activated by lasers, genetically induce extra eyeballs growth, or genetically engineer laser eyes like Superman? It's a really fun question to read, by the way. Lasers.
Wow, that is a really fun question. Well, optogenetics, I see the root opto in there, which I know has to deal with vision. So if I'm narrowing these questions down, in answer A, I don't see anything about vision, so I don't think that it's going to be that one. But toss up between can they genetically induce extra eyeball growth or engineer laser eyes like Superman, that's a tough one, because both of those sound like those would be incredible feats of scientific achievement.
I can find uses for both of them, absolutely.
Yeah, but I'm going to say C.
Genetically engineered laser eyes like Superman. All right, let's see.
Go for
No, it is genetically engineered neurons to be activated by lasers.
By lasers. Oh, so that must be then that it's more about, not about the eyes themselves, but about vision and how if lasers, that's if something sees the laser or something, then or that triggers part of that vision, part of the brain, then that must be how that's connected.
I read a question like this and I think, oh, I have something to learn after this.
Yeah, I was thinking about that in a totally different way.
All right, number 5. What scientific disciplines are Sheldon and Amy from the Big Bang Theory working on? Is it A, string theory and neuroscience, B, string theory and mathematics, C, math and chemistry, or D, history and geography?
I remember this one. I've seen some of this show, and I'm going to go with the answer A. Sheldon works with string theory, and Amy is a neuroscientist.
All right, here we go. Yes, you got that one right. Okay, I don't have anything to say about that show. I hear it's good. All right. Psilocybin has recently been approved as as breakthrough treatment for which medical treatment? Athlete's foot, COVID-19, major depression, or boredom?
Well, since we are talking about the brain tonight, I'm going to go with C, major depression, as that's more of a mental condition than something like athlete's foot.
I don't know. I got a lot of people who think that boredom is connected to just not having anything to think about.
It could be boredom. Yeah, I was kind of thinking, well, it could be C or D, depression or boredom. But the way that the question is phrased about how it's a breakthrough treatment for which medical condition, I'm going to go with major depression, since that has the word major in it, and boredom is more of a normal day-to-day thing for everybody.
Not exactly a medical condition.
Yeah.
All right. And yes, you are right. Major depression. So interesting event on that as well. All right. Number 7. How many neurons are there in the human brain? This goes back to our first question. How many stars in the galaxy?
Yeah.
2.8 million and declining daily. 85 billion, 1 trillion, or just too many to count. It's not going to do it.
Well, yeah, this does go back to our first question. And I was thinking when I said that question that there are at least hundreds of billions of stars in the Milky Way. And now I know that there are more neurons in the brain than there are stars in the Milky Way. So I'm going to rule out A and B because those are less than hundreds of billions. But too many to count doesn't seem very scientific. So I'm going to go with 1 trillion.
Yeah, I feel like we could do better than too many to count. That seems like somebody has a project.
Some kind of estimation. Because too many to count could be 2 trillion. It could be 200 billion, trillion, million. Who knows?
Yeah, we're definitely a little bit more precise than that, even though 85 billion is still pretty hard to count.
Oh, well then I guess I overestimated the number of stars in the Milky Way.
All right, 2 things you're going to have to go back and check and learn more
I
about.
guess so.
All right, question number 8. What percentage of the brain does the average person use on a daily basis? 10%, 38.6%, 100%, or most days we only use reflexes?
This is another good question. Well, right away I'm going to rule out D because like the last question, that's not a very scientific answer. And we're a science museum here. So A, I have heard that that is a common saying that people only use 10% of their brain, but I've also heard that that is a myth. So I'm also going to rule out A. So the question now that I'm coming down to is it 38.6% or 100%? And I'm thinking that maybe we're using all of our brain, but not actively in the same way all the time. But 38.6%, you know, that could be an indication of, well, it's more than 10% and maybe what we're actively using, but we're not actively using the whole thing on a
All right, what's your final answer?
Final answer, I'll go with B since it's in the middle.
All right. Oh, 100%.
Yeah, that's, I should have gone with my gut there, with my brain there.
All right, number 9. Approximately what percentage of your brain is white matter? White matter is the brain wiring that connects those regions, all the regions. Is it 10%, 25%, 50%, or 75%?
Another good one. Well, I don't think that it's 75% or 50% necessarily, because if it's just the wiring, then that would take up less volume than I've heard about gray matter, and I would think that that takes up more. So I'm narrowing it down to A or B, and I'm going to go with B, 25%.
All right, and
50%.
50%. Yeah. Again, things I don't know about either. All right, last question. What percentage of water is your brain? Is it 28%, 49%, 65%, or 73%?
Another question all about the percentages. Oh, these are tricky with these precise numbers here. I like this. This is fun. And I bet a lot of people out in the audience are thinking this over carefully and are going to get this one right too. So I typically, when I look at multiple choice questions, I don't usually think that it's the lowest or the highest number when it's a percentage like this.
Okay.
As for what percent water, well, I'm going to say 65%, because I know that the rest of the body is maybe somewhere around there too. It's more than 50% water. So I'll go with 65%.
All right. And 73%.
Oh, wow. Okay.
So those numbers actually look like they got pulled from some of these other, the skin, it contains 64%. Bones have 31%. Lungs about 83%. We have an average of a lot of water, but specifically the brain, 73%.
Great. Well, I learned so much already, and I'm so excited to learn more with Dr. Mazzucato tonight.
Thanks for coming, Courtney. I'll figure out, you get a cookie anyway, because you're just brave for answering questions that you didn't know about. So thanks for learning along.
Thanks, Anne. Looking forward to learning more. Bye-bye.
All right, and now I'd like to introduce our speaker. Dr. Mazzucato is a theoretical physicist by training. He obtained his PhD in theoretical particle physics at CISA in Trieste, Italy in 2005, and worked on string theory and beyond the standard model physics at the Department of Particle Physics at Tel Aviv University, and as a visiting researcher at the Rocca Institute of Physics at Hebrew University. He was a member at the Simmons Center for Geometry and Physics and a visiting scientist at the Kavli Institute for Theoretical Physics in Santa Barbara. He began his neuroscience research in 2012 at the Department of Neurobiology and Behavior at Stony Brook University. He was a Schwartz Fellow in Theoretical Neurobiology and, since 2014, an NIH-funded principal investigator. He was an associate research scientist at the Center for Theoretical Neuroscience at the Zuckerman Mind-Body Behavior Institute at Columbia University. Luca Mazzucato is currently an associate professor at the Departments of Biology and Mathematics and a member of the Institute of Neuroscience. Take it away, Dr. Mazzucato.
Hi, everybody. Thanks so much for inviting me to the Science Club. I'm very excited to be here and to share with you some results from my lab. So I recently moved to Eugene in 2018. So I started my lab here at the University of Oregon at the Institute of Neuroscience. And what I want to talk about today is something that it's really dear to me, and it's the origin of time in the brain. And so if we think about the origin of time in the brain, there's many ways to address this question. And so one way you could address this question is to sit down at your desk and think very hard about this problem using some kind of introspection, using some metaphysics or ontology. And you can come up like with some answer like the German philosopher Immanuel Kant once said, so that space and time are pure forms of sensible intuition, and in particular that time is the awareness of your inner self. So I let this sink for a while, and then I want to tell you instead of a different way of approaching the problem, which is what we do as neuroscientists, which is particularly suited for this kind of COVID-19 pandemic times. So what we do as neuroscientists is we sit down and use our laptops, and we tend to watch kind of YouTube videos all day long, and we watch videos of cats and dogs and like funny animals and for hours and hours at a time. And so this is what we do in the lab. And then after watching all these videos, we come up with hypotheses and conclusions about how time is represented in our brain. And so today I want to share with you some of our results of these investigations. And in particular, I hope by the end of the talk, I will convince you that this approach of watching videos is as fruitful as, hopefully, as continental philosophy. So let me start with the lab's favorite video. And this is a video of a mouse. And so this is a video of the mouse where the mouse is engaged in some spontaneous and self-initiated behavior. So as you can see, the mouse is exploring this walnut and really wants to get the walnut inside their own house, which is this hole in the ground in this particular case. And so I want to highlight a few features of the spontaneous behavior of the mouse. So the spontaneous behavior of the mouse starts from some very fast movements. So fast movements that could be like whisking, moving their whiskers, sniffing, wagging their snout, or like moving their paws very fast, right? Or like biting the walnut very fast. And all these kind of actions or movements are typically occurring at a sub-second scale, like a tenth of a second, like 100 milliseconds. And so these are like very fast time scales of mouse movement. However, we like to think about behavior as a kind of a language. And then, let me play this video again. And so, and then we can see that there are multiple time scales of behavior emerging from these movements. And so, on top of this very fast time scale, there's another time scale that's like an intermediate time scale, which would be, let's say, about 1 to 10 seconds long time scale, in which you concatenate the syllables, these fast actions, into action sequences that have a behavioral meaning for the animal. They could be, for example, walking out of the hole and grabbing the walnut and bringing it back, or hiding. It's kind of intermediate 10 second long, 1 second, 10 second long time scale. At the same time, the whole purpose of the animal, like his overarching plan, exists on a much longer time scale than this, which is at least the length of the video, that's like a minute, but in general could be much, much longer, and for the animal to be able to forage for food successfully. And so this is like, if you want to think about behavior again as a language, this is going to be how you put together words into longer sentences. And so already from this very simple YouTube video, you can see the emergence of an extremely complex structure in the behavior, which involves several multiple time scales that coexist simultaneously. So going from 100 milliseconds, like a tenth of a second, to seconds or tenths of seconds, all the way to minutes. And, in the case of humans, like in the case of my lab, the typical time scale is 5 years to get a project done. Like this one I'm going to talk about, say in Soviet Union, they used to think in terms of 5-year plans and so on and so forth, or even longer plans, right? So we have always like this kind of multiple time scales of activity all coexisting at the same time. And so as neuroscientists, we make like a crazy hypothesis, and we want to understand this behavior in terms of neural activity. So we believe that behavior is generated by neural activity. And in particular, what I'm going to talk about is neural activity somewhere there in cortex in our brain. So the story, in fact, is much more complicated than this. And in the last few years, we realized that not only is behavior generated by neural activity, but neural activity reflects behavior as a backdrop, as a proprioceptive feedback from movement. So there's a very complicated interplay between movements that are generated by motor commands and like reflected post hoc in neural activity. And so if you want, this is like some kind of a chicken and egg problem that's very complex and exists at all these different timescales. And so this is what we try to address with the research in my lab. And so before we move forward, I want to just spend a few words about the lab and our collaboration. So this is my lab at the University of Oregon, and these are the people in the lab. And the work I'm going to talk about today is mostly due to an amazing collaboration between theoretical neuroscientists and experimental neuroscientists. So in particular, Stefano Recanatesi and David Weyrich are the two theoretical neuroscientists. And Ulisse Pereira at NYU is also a collaborator, theoretical neuroscientist. And Zach Meinen, Mazu Murakami, and Fanny Kazet are experimental collaborators in Lisbon that developed this experiment I'm going to talk about. And of course, our experimental subjects, which are going to be rodents in today's talk, but could also be primates or humans. And so, in general, I want to just give you an idea about like our enterprise in the lab is a truly kind of global collaboration involving people from the US, from the East Coast and West Coast, and in Asia, like in Japan and Europe and Israel. So it's a truly global collaboration that is addressing this particular question, like this how time emerges from the brain. So. Okay, so now what's the plan of the talk? What I'm interested in terms of emergence of time from brain activity, we can look at it from three different points of view. So the simplest point of view, or the first point of view historically that was addressed in neuroscience is called classical conditioning. And so is the idea of the Pavlov dog, or in this case, this super cute Pavlov cats, if you want. So is the idea to link events in time. When event A predicts event B, then you create a notion of time as an order in which these events occur. So whenever you ring the bell, you learn that you can get a little treat. And so that's how you start to think about time as the flow of time. So, but there, I want to draw your attention to something kind of a different point of view about time, which is, again, I'm playing again my favorite YouTube video, the mouse. So, is the idea that because we can control our movements, we can create action sequences which concatenate our own movements in time. So this is like a active exploration of time as opposed to a passive exploration of time by seeing external signals, right? External stimuli. We can actively explore time by creating this flow of movements or these action sequences. And then once we can create these action sequences, then we can also compress them or dilute them, the same action sequence, so we can control the timing of our actions. And so, for example, mice perform the same action very fast, but sloth perform this action very, very slowly, like in this awesome movie that probably most of you have seen. And so in the first part of the talk, I will focus on how, on this amazing experiment that was done to study the origin of these action sequences. So how does the brain generate these action sequences during behavior? So let me be more specific and get into some kind of the details of the neuroscience of this kind of experiment. So what we're considering is spontaneous animal behavior or self-initiated animal behavior. So what the animal does spontaneously, as opposed to what they would do if they're instructed to do so by the experimenter. So spontaneous animal behavior is extremely complicated. And I'm showing you here some figures from a paper by the Bob Data lab at Harvard. That was a seminal insight into this problem. So one can use machine learning or artificial intelligence techniques to classify the spontaneous behavior of animals into syllables, just like we were talking about before, into very small actions that are the syllables of behavior. And so in particular here, I can show you 3 examples of syllables that would be a rear pause. So whenever the animal is just moving up, right, standing up on their hind legs, or a dive when the animal is like going down. This would be the second syllable, or another syllable could be locomotion, where the animal is just moving forward in a straight line. And so now there's many sources of variability in behavior, and in particular, what we just saw is that there's many actions or syllables that the animal can take at any given time, right? So this is a very large number of potential or possible actions the animal can make. But the problem is even harder, because given a set of actions, the animal can concatenate them in many different ways to perform a very large number of action sequences, right? The animal can do a rear pause, a dive, and a locomotion, or a locomotion and a rear pause, a dive, and a rear pause again, and so on and so forth in many different combinations. And then there's even a third source of variability, which is what I'm mainly going to focus on in this talk, is that even given this large lexical complexity in behavior, it's even more complicated than that, because every time the animal performs the same action, for example, every time the animal performs a dive, that's shown here in blue, and this is like a color-coded action sequence. Whenever there's a blue interval, this represents a short time interval when a dive was performed. Whenever the animal performs the same action, they can perform it very fast or very slow, or at any time timing in between. So there's a huge variability in the temporal dimension of behavior. And so because of this huge lexical complexity and temporal complexity, this seems like a hopeless problem to solve, to try to figure out how this temporal structure in behavior arises. And so what we set out to do was to try to control some of the feature in behavior. And in particular, we decided to control the lexical complexity in behavior, or the spatial complexity, by designing an experiment where the animal was performing a fixed set of actions in a fixed order. So the animal could perform one single word if you want, but they were free to perform it at any given time, so that the behavior was still self-initiated. So we fixed, or we controlled for this lexical complexity with this combinatorically large lexical complexity in behavior, which would be hopeless to attack, or at least that I wouldn't feel able to do. And we focused on the temporal dimension, so the emergence of these action sequences with complex timing. And so, in fact, I hope I convinced you by the end of the talk that this allowed us to make actual progress in the quest about this the structure of time. So let me describe a little bit of the experiment, because it's a very cool experiment that was done by Mazza and Zach. And the theory part of this project was done by Stefano and Ulysses. So many of you might be familiar with the so-called Marshmallow Experiment. So this is a famous psychology experiment from many decades ago, in which, you know, there was a kid that was left sitting at a desk in a room by himself. And so they had a marshmallow in front of them, and they were told, well, you can either eat the marshmallow now, or if you can control yourself and you're not going to eat the marshmallow now, you can get 2 marshmallows later. And so, okay, so there's a lot of baggage with this experiment. So, and what, I'm not going to talk about it. What Mazza and Zach did is that they developed the rodent version of the Marshmallow Experiment. And so let me walk you through. So this is the schematics of the rodent version. So we have a rat. The rat is in a box, and the box has 2 ports where the animal can poke their nose in. The animal just pokes the nose into the wait port, the waiting port, and that's how the animal decides whenever they want. The animal decides they want to engage in a trial. So we call this particular one realization of this sequence a trial, an experimental trial. So when the animal pokes into the waiting port, which is the same as the kid sitting in the chair in the room, then a tone signals that, okay, trial is on. The experiment starts, and then the animal has to wait with their nose poked into this waiting port, and they have 2 options. So they can either wait for a very long time for a second tone to play that happens at random times. And then if they hear the second tone, they can poke out of the wait port and then move to the second port and collect some reward, like a big reward, like many marshmallows, because they were very patient, right? So, well, marshmallows for rats in this particular experiment is just like drops of water, you know. They have low expectations. But at any time, the animal has a second option that they can take at any time. If they feel impatient while they're waiting, they don't want to wait for this long time to get the second tone. They can just poke out at any time from the wait port and poke into the reward port and get a very small reward. So they can be either patient and get the large reward, or impatient and get a small reward. So notice that in this experiment, the action sequence the animal is making in both conditions is exactly the same. But because this is self-initiated, the animal can perform these actions at any time they want. So this is what I was talking about before. So we control the lexical variability in behavior, and we only look at the temporal variability. And so I want to give you an example. I want to show you one experimental session, how large the variability in spontaneous, the self-initiated behavior is. And so to the left here, I'm drawing a diagram of this temporal interval. So every vertical bar here describes one experimental trial, and the length of these vertical bars, they could be dashed or full, they describe how long the animal is waiting into the wait port, with their nose poked into the wait port. And so you see that sometimes the animal waits for less than a second, then on the third trials, then the third trial, they wait like 3 seconds, then 5, then they go down to 1, and then they go up to 10 seconds some other time. And so sometimes they're patient, so that's a full line. Sometimes they're impatient, that's a dashed line. So there's a huge variability in behavior, even if, there's not much the animal can do, but still they have this huge variability. So we're going to focus on this variability in behavior. And so we're going to ask the following questions. So first of all, how are actions represented in the brain? So each of these actions the animal is performing, is there a representation in the brain for each of these actions? So how does the brain generate these action sequences driving behavior, the spontaneous behavior? And how does the brain generate these variable waiting times, or this variable action timing? So these are the 3 questions we're going to focus on. And in order to get there, I have to make a slight detour and give you like a one-on-one of like what neural activity looks like. So, so let me back up for a second and pause on the experiment to show you how we actually extract a neural activity from the brain. So what you see here to the left is like a view from the top of a mouse brain. So this is the front of the brain here. And then to the right, you have the back of the brain, right? And these are the two hemispheres. And so I put a circle, a pink circle on the specific brain area we're going to record from. We're going to extract neural activity from, which is a part of the frontal cortex, which is called secondary motor cortex or premotor cortex, which is believed to be related to intentions. And we will get there in a second. But first, let me give you like a quick tour of how we actually measure brain activity. So we insert electrodes into cortical tissue. So this is supposed to be, this cylinder is supposed to represent cortical tissue, and every triangle is supposed to represent like a neuron around the electron. So this electrode is picking some electrical signal coming from all these neurons around it that send messages to each other all the time. And so we high-pass filter, we filter this electrical signal in order to identify from this combined signal the fingerprints of each single neuron around the electrode. And the idea is that neurons communicate with each other by emitting some kind of a digital signal that's called an action potential or a spike, which is represented here by like a sudden increase in the electrical signal picked up by the electrode. This is super fast, like 1 millisecond. And the crucial issue here is that every neuron has their own particular fingerprint of their spike. So what I'm showing you here are spikes from 3 different neurons, a red one, a green one, and a blue one. And you can see that each neuron has a certain height or width of their spike, such that we can identify within this electrical signal which neuron spiked around the electrode. And so then we can represent neural activity from this particular patch of cortex in terms of, let's say, schematically here, I'm showing you the neural activity from 4 neurons, red, green, blue, and black. And so on the horizontal axis, we have time, passing time, and every vertical bar it's a tick that represents when that neuron emitted a spike. And so at the end of the day, this is like all amazing experimental work that's been done by our experimental colleagues. We are handed a bunch of spike times. These are the spike time, the times at which every one of these neurons emitted their own spikes. And you see these are all simultaneously recorded neurons that we have recorded from the same electrode of the same electrode bundle. And so then we want to study the language that these neurons talk to each other. So this is very complicated, and neurons talk to each other all the time, even if nothing is going on in the brain. So it's called spontaneous brain activity. And to give you an idea of the complexity of this neural code, of this chatter, in non-stop chatter that neurons are like using to communicate with each other, in the next slide, I made a little sound clip in which now we have one raster plot. So this is called raster plot, and you see a couple of those. And let me walk you through. So a raster plot represents time here, the passing time on the horizontal axis from 0 to 1 to 5 seconds here. And every line represents the activity of one neuron. For example, the first line represents the activity of neuron 1, one particular neuron recorded that emits some spikes at these particular times where there's a vertical bar. And then the second line represents neuron 2. Neuron 2 is super active at the beginning, then kind of shuts down. And then as you go up, the one but last neuron here is kind of active a little bit and then becomes super active at the end or so. So to give you an idea of what this feels like, I made a sound clip in which I gave a different blip to each neuron. So the first neuron has a very low tone, like bloop, 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 up to the top neuron that has a very high-pitched tone, like doo, 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 doo. And then I play them all together. And so now you hear what's going on in the mouse brain. Okay, so I don't know if I hope you could hear the sound. So this is the kind of signal that everybody in the lab is trying to make sense of. And so now we go back to the experiment, and I remind you, this is the experiment we're looking at. We have a mouse poking into the wait port and then getting out of the wait port when they get impatient or they hear the other tone, and then moving to the other reward port and collect a reward. And so we're going to record from the secondary motor cortex, motor cortex of rats. And I'm going to show you some raster plots again. So these are raster plots recorded from 2 different trials. So two different times that the animal was performing this action sequence. So on top, there's the first trial, and on the bottom, you have the second trial. And this is the same notation as in the previous slide, in which on the horizontal axis, you have time here within the trial. And every line represents the activity of one particular neuron that was recorded from these electrodes. So we have 12 neurons here in both trials. And you see that there's every spike is a black dot, right? And so you see that there's some kind of pattern of spiking activity appearing in both plots. And so if we sit down long enough and we stare at these plots for a long time, we start to hallucinate some patterns. And we kind of start to make hypotheses as to what the neural code is in this experiment. And then we can use some techniques, some machine learning techniques to extract these patterns. So for example, what do I mean by patterns? So I highlighted here with a gray box a pattern of neural activity where neurons 11 and 12 fire a lot, and the other ones are kind of silent. See, and this happens in this trial, and also kind of happens in this trial as well, right? So neuron 11 and 12 fire a lot of spikes in this interval, but the other ones are kind of silent. Then there's another pattern that I highlighted with the red boxes. And so this is where neuron, I don't know, what is this, like this is like 7 maybe, and 11 fire a lot, and the other ones are kind of silent. And you see that this happened in both trials. So what we do is we use a machine learning technique that's called a hidden Markov model, which is the same technique that's used in speech recognition, like, you know, on your phone. To extract syllables from your speech, we extract syllables of neural activity from this time course for this time series. So it's kind of the same, exactly the same idea. And so when we use this machine learning algorithm, we can segment the neural activity in the trials according to these syllables, these neural patterns. And I highlighted here in these 2 trials patterns that are detected by these algorithms in different colors. And you see that the sequence of colors is the same in both trials, even though every trial has the syllables in each trial last for different intervals, right? It's the same thing when you speak. You can speak faster or slower. The syllables are always the same, but they are compressed or stretched. And that's why we use this kind of algorithms. So I want to represent now the whole experiment in a very convenient, compact way. And I do this as follows. So these are 6 trials now. I showed you the first 2 in the previous slide. Now I'm showing you 1, 2, 3, 4, 5, 6. I want to take an abstract view of these trials. Now that I have a sequence of syllables or neural patterns, I can forget about the neural activity underneath. I can only study these sequences of patterns now. And now the sequence of patterns is just a sequence of colors, because that's how I represented it. And now I can take all the trials in one experimental session in 1 hour of recordings, if you want, and put every trial one next to each other. So the plot on the right shows all the trials in this 1 hour of recording. There's 180 trials. Every line in this plot represents one particular trial, and the colors on each line represent the sequence of neural syllables, neural patterns they extracted with the series mechanism in that particular trial. And I align them all one below the other. And so, of course, trials are self-initiated, so they have different lengths, and these sequences have different lengths, right? And so you see that, in fact, this is reflected by the fact that they're not all aligned at the end, even if I align them at the beginning, right? And to have a better way of visualizing this, I can do a trick now. I can just stretch all the trials so that I align them to the same behavioral events. The behavioral events are the poke in, the wait, and the poke out, and then when they go collect the water. So I'm going to stretch all the trials to get them all aligned at the same length. And now this is what the stretch sequences look like. And the sequences are aligned in the following way. So I stretch all the trials such that all the times where in cross trials where the animal was poking in are now all on top of each other. All the times where the animal was poking out are all aligned on top of each other. And all the times where the animal was getting the water reward are aligned on top of each other. Now, if I present the sequences like this, you see this striking result that the neural pattern sequence is the same in all trials, even if all trials were very different. So in particular, let me draw your attention to the fact that before the animal was poking out, wanted to eat their marshmallow, right? So we can predict the time where the animal is going to perform this action, this poke out, by the onset of the red pattern. So whenever the red pattern comes on in the motor cortex, we know that very soon after, 200 milliseconds later, the animal is going to poke out. So with this algorithm, we can read the animal's mind, and we can predict what the animal is going to do next, right? So this is kind of one of the results of this project, in which now we know what the neural code for action sequences in this complicated self-initiated task is, and we know that we can predict the animal's actions from these neural patterns in motor cortex. Now, because we're theoretical neuroscientists, this is not enough for us. So it's not enough for me to be able to tell what the statistical structure of neural activity is. I want to build a model that generates the same activity. So this is kind of an artificial intelligence point of view. I want to build a neural circuit now that produces the same activity that I observed in the motor cortex. In particular, I want to produce neural circuits that generate the same action sequence, but however, where all of these different intervals of actions are variable in time. So across this kind of scales from 1 second to 10 seconds. And so I want to introduce you to some kind of fun tool that we use in computational neuroscience. So these are called neural networks or artificial neural networks. And so the idea is the following. So, we start with this kind of brain tissue, these cortical circuits. And cortical circuits, here you see this rainbow picture of a cortical circuit. These are super complicated circuits made of neurons that have different morphology, different shapes, complicated connectivity, receptors. And so, but because we're all physicists, so physicists operate according to the principle of a spherical cow. And so here we use the principle of spherical neurons, in which we kind of forget about the awesome biological complexity of these cortical circuits, and we just approximate them as a graph, a network. Here to the bottom, an artificial neural network in which every node in the network is a neuron, and every edge represents a synaptic connection between the neurons. And so we can simulate now these artificial neural networks according to some simple mathematical algorithms. And what I'm showing you here is, for example, I picked 3 neurons from this artificial neural network in blue, red, and yellow. And so what I'm showing you here, I initialize the network with some random initial condition, and I let it evolve. And so these neurons have some kind of time varying activity, and then they reach a steady state, a stable state. And so this stable state we call a tractor. Because if you start close to this particular state of activity, where neuron yellow, red, and blue have these particular values, if you start close to them, you are attracted to that particular state. And if you perturb this attractor, if you perturb the neural activity a little bit away from this state, you go back to it. But if you perturb it too much, then you can move away from it. So we represent these attractors in terms of potential wells. Imagine you have some kind of a rugged landscape, and neural activity is like a ball that's like navigating this two-dimensional rugged landscape. So the ball is gonna kind of stop a little bit around some wells, some potential wells, and every potential well represents a different attractor. So for example, now we're stopped here in this particular potential well. This represents this attractor. But we could move to another potential well, which is represented by another attractor. So neural activity, which spends some time in this particular attractor, and then it jumps into another attractor. So the idea is that these attractors in the artificial neural networks represent, in a very specific, precise sense, the syllables of neural activity that we found in motor cortex. So these attractors represent the actions, the intention of the next action the animal was going to make. And so concatenating these attractors, or if you want, for neural activity to explore of a sequence of potential wells, this represents the action sequence that generates the behavior. And so, in particular, we can now compare the activity in these artificial neural networks with the activity that we found in the brain, in the motor cortex. And I'm showing you here, for comparison, to the left, we have, I performed this kind of hidden Markov model syllable extraction on the artificial neural network, the same exact syllable extraction I did on the motor cortex data. And it really looks, so it has the same exact statistics. And so we can compare it and make, quantitative comparison between them. And the idea is that now we identify the syllables from motor cortex neural activity as attractors. And these action sequences or like sequences of intentions of the next action as sequences of attractors. But because we have a model that describes the microscopic circuit, we can now ask mechanistic questions. We can ask, what is the mechanism that drives the action sequence, that drives the decision to perform the next movement or the next action? And it turns out that in the model, the mechanism that drives this transition is noise. So the fact that the same action lasts different amount of times across trials, so every time you perform the spontaneous action, it takes a different time to perform it. This is due to noise. And this explains very, very well the temporal variability that we found in the behavior of this in this Marshmallow experiment. And so let me give you a quick summary of what we got so far. So far we found that motor cortex activity can be described by these sequences of neural attractors. Every attractor predicts what action the animal is going to perform next. And this transition, this large variability in the timing of action is driven by noise. And so I want to spend the last 3 minutes here, 5 minutes of the talk by now explaining some applications of this framework, this theoretical framework that we have. Once we have this framework in place to describe sequences of actions, now we can ask questions like, what controls the timing of actions? Can we speed up or slow down the timing of action? So can we compress or stretch this the flow of time in our movements? And so now if we work out a little bit of math, and this was done, I was in collaboration with David Wyrick, a student in the lab. So now we can study what parameters determine the transitions from one attractor to the next attractor. And it turns out that a mathematical analysis of this artificial neural network show that this parameter is the height of the barrier separating the attractor. So if I want to move from attractor A to attractor B, the probability of the transition, it depends on the height of the barrier. So the higher the barrier, the slower the transition. If I find a way to shrink the barrier separating these attractors, then I have a faster transition. So you can have two different states. A state in which I have high barrier, and so I move slowly, or I have slow reaction times, and a state when I lower the barrier, so I move much faster. So like a sloth would be the high barrier brain, and like a mouse would be the low barrier brain, if you want. But I can also study a question which is, can the brain control, the same brain, now not two different animals, right, but the same brain in two different brain states, if you want, can we control the timing of actions? And to study this question, they will look at visual responses of mice that were running on a ball. So this is another experiment in which there was a mouse that was sitting on a ball, and the mouse could stay still or could run. And during this time where the animal was sitting still or running, whatever they liked, so this is like spontaneous behavior, the mouse was shown some images through a screen in front of the mouse. And so we analyzed data from the visual cortex of mice. Now this is in the back of the brain. This is a sensory cortex that gets the inputs from your eyes, from your retina. And this is from the Allen Institute up in Seattle. And so we asked the following question. So is there a faster reaction to visual images when the animal is running compared to when the animal was sitting? And in fact, this is exactly what we found. So we found that when the animal is running, and here in red, you see the response to images from visual cortex neuron, there's a faster response to images when the animal was running compared to when the animal was resting. And we developed a model that explains then this in terms of changes in these potential barriers of attractors in the visual cortex. And so, in fact, we claim that this is a very general phenomenon that now applies to both, motor generation, visual cortex, or previously we collaborated with Stony Brook University. I showed that the same framework applies to taste processing. So explains the fact that when you expect some stimulus, you recognize it faster compared to when the same stimulus is shown to you by surprise. So we claim that this is like a very general phenomenon that describes brain activity. And so let me wrap up now and summarize what we talked about. So how does the brain represent actions? So every action, the intention of the next action is represented by an attractor. So we can read the animal's mind by detecting the attractors and predicting the action before the animal decides to move. And we can generate action sequences by now concatenating attractors in this landscape of potential wells. And so the transitions between attractors are generated by noise. And this explains why in the spontaneous behavior, sometimes you move fast, sometimes you move slow, even if you perform the same action. And at the end, I showed you that, well, now we can manipulate the speed or the flow of time in these movements by changing the barriers between attractors in the neural activity. And this explains the effect of expectations or running or other effects like attention on the processing of incoming stimuli or on movements, outgoing movement. And so, so I didn't talk about how we link events in time and create memories or like Pavlovian conditioning, so because this is a whole other talk, but you can look at papers on my website about that. So I want to conclude the talk by kind of speculating why it's interesting or important to have this timing variability in the brain. So why do we have this mechanism such that spontaneous behavior generates this very, very, very kind of complicated timing of actions? Well, what you see here to the left is like a very complex sequence of actions that this bear is making when they're going fishing, but it has perfect timing. So they must know exactly the right time to kind of open their mouth and grab the fish that's swimming upstream, the salmon that's swimming upstream. But bears, I mean, the only thing you see in videos is this kind of amazing bears that always catch the fish, right? So, but how do they get there? Well, and now the way to get there is because when they start and they're like, juvenile bears, they really have no idea how they're gonna get this time right. So they know the sequence of action they want to make. They know they have to open their mouth and kind of catch the fish. But it takes a very long time for them to get it right. And so then it's really useful then to have a mechanism that explores random timing in an action sequence that you know very well in order to get it right, to learn how to get the timing right. And so this is a mechanism to explore the optimal timing in a stable behavioral sequence whose actions you already know. And okay, so this is a possible speculation on why this mechanism could be useful for cognition. Let me go back and thank everybody in the lab, in particular what I talked about today. This is work by Stefano Canatesi and David Weyrich, and my collaborators at Champalimaud Center for the Unknown in Lisbon, the Many Neurons, so with Masa and Fanny, and Ulysses Pereira at NYU, who were the other theorists on this project, and of course the rodents that were the subject of the experiment and our funding agency. And thank everybody for listening, and I'm looking forward to hearing your questions.
All right, thanks for a great lecture on the reaction times in the brain. My first impulse is like, I work with youth, and we sometimes play games where you mimic animals, and it's really easy to figure out. You're talking about those sequence of how long the duration is and how long somebody play acts of walking can be the difference between a horse or a cat. And those are things that we kind of already observe when we look at animals. So it's really fascinating that you're kind of parsing those down to really understand that on that brain level. All right. Also, double thumbs up for all of your videos. I know that I love that bear one. I really do. Every time that bear is just fantastic. So a couple of questions did come up. In terms of can you go into more about what is the potential application of your research to human brains, and are we able to think about training ourselves to have faster reaction times? Can you go into that at all?
Well, yeah. So all of the neuroscience work, most of the neuroscience work is kind of starting from some question about some mental illness or some application to some disease. And so, for example, in this particular case of motor generation, motor sequences, one potential application, which I'm not working on, but it's motivating all of this work, is, of course, the idea of understanding the origin of movements to be able to develop some treatment for disease that involve motor control. And in particular, I would think about Parkinson's disease, which is affecting movements and sequence of movements and motor control. So this is one of the main applications that I can see going forward. So the idea would be that once you understand the neural code for the motor cortex is using to generate the action sequence, in the case in which it doesn't work anymore, you can try to restore it with some for example, this is a bit far-fetched, but some brain-machine interface that would intervene and stimulate cortex when there's a failure of acting. Or, for example, in case where there's a stroke, a patient with stroke whose part of the motor cortex was damaged, they lose the ability to move. And if we understand the way the brain is generating these complex movements, then we can think about ways to restore the function in stroke patients. So these are two clear directions that we could go once we really understand how this works. Well, this is in mice. You have to understand how this works in monkeys then, and then think about humans. So, but these are two particular applications.
Right. Okay. Yeah, they're this question says, I think it's just a little bit more to extrapolate a little bit more about what you were saying. Do you see these findings applying to patient care with those neurodevelopmental or behavioral uniqueness?
Sorry, neurodevelopmental?
It says, how do you see these findings applying to patient care with those neurodevelopmental or behavioral uniqueness? I mean, you were talking a little bit further down the way, but
yeah. So I don't have any experience of working with patients, unfortunately. So I don't have anything specific to say about this, except that I'm looking forward to starting collaborations with colleagues who work with human patients to address these problems. And there's some of them, University of Oregon in particular, that I'm thinking about.
Okay. So you're talking about how all of this is instigated by noise in the brain. So does the noise in the neural network correspond to something real in the brains? And is it something that we can speed up or slow down?
Yes, so this is a great question. So as a theorist, I'm really obsessed with this question about noise. And I can speculate a little bit about this. So we have very specific mechanism in mind for explaining where this noise comes from. And we're designing experiments to test what kind of noise. So let me give you a couple of examples. So one example is the fact that when these neurons discharge their spikes, their action potentials, all these discharges are noisy, so they're not precise. So the timing of the spikes is not precise. So sometimes there's a failure in release of some chemicals at the synapses that make the spike occur at a slightly different time. And so this generates naturally noise. And so if the signal propagates within a circuit that we believe is a circuit involving motor cortex and some other brain area, so this noise gets amplified in some way that generates this movement, this temporal variability. So this is a possibility. There's an alternative model that we're also exploring, which I think is super cool, that involves chaotic dynamic. So instead of having noise in the microscopic discharge of this mechanism at synapses, there could be a complex temporal dynamics in some other part of the brain, let's say some other part of the prefrontal cortex, because the neural activity is always dynamic. The activity in some other part of the cortex could be read by the motor cortex effectively as noise if this is not related to anything that the motor cortex is doing. This is some completely orthogonal calculation that some other brain area is doing. And in particular, it's a chaotic dynamics that this other brain area has. So chaos in another part of the brain could generate the noise that is needed in motor cortex to drive these transitions. So these are two alternative scenarios that we are actually currently exploring, and we're designing new experiments with Zach Maiden and their lab to actually test between these alternative scenarios.
Interesting. Have you or any of your colleagues collaborated with musicians as you've studied these mice brains?
This question. So these questions are like about things that I'm really thinking about. So yeah. So in fact, one of the projects we're working on. It's very hard to make progress on this project, but is to compare the behavior in mice or in animals in general, so the spontaneous behavior that I talked about, with spontaneous behaviors in humans. Now, we needed some hook on the spontaneous behavior in humans such that it was not too complicated, but still included some complex variability. And so what we thought about was that jazz improvisation would be the natural kind of analog in humans of this, because in jazz, you are kind of improvising on a script. So there's a structure of a song, but every time you play the song, you slightly change it. And so the idea is that kind of jazz improvisation would be this lexically constrained but temporally variable, if you want, or slightly lexically variable variation on a stable behavioral sequence. So we're trying to explore or compare these two approaches: spontaneous behavior in mice and improvisation, musical improvisation in humans. But I don't have anything to say at the moment about it.
So kind of back to your graph, when that red line you were talking about, that would be one instrument or one note, and you know that would happen, then something would follow in the jazz sequence. That's your parallel.
Yes.
Cool. All right, interesting. Another question that came up says, does random timing variability broadly change across the lifespan of the organism, or does it decrease as behaviors become habitual?
So that's a great question. So I haven't worked on that personally, but I can say something a bit vague about it. And so one of the ideas I presented at the end with the bear video was that there is a change in variability that comes with learning, and we turn in some kind of exploratory behavior in a naive animal, in a naive condition when you first approach the problem. As you first approach the problem, you need to explore the time, so you have a lot of variability. But then as you learn, you're going to constrain this temporal variability into the optimal timing, so that if you perform this sequence many times, you finally get it right. And so you, with habit, if you want, then this becomes perfect. And so the temporal variability goes away. So this is about when you learn the right timing for the same temporal sequence. Now, it's a great question in general to ask in this exploratory spontaneous behavior. Now, when you're not learning some particular sequence, but when you're just acting spontaneously, is there a difference in this variability between juvenile and older subjects? And so I haven't thought about that at all, actually. And so this is a very good question that thanks for bringing it up. So I'll try to think about it.
Well, on a similar note, what about relearning something? Because we've all learned actions and then time has gone by, we've forgotten them, and you started again, you have to think, I'm at the beginning learning this. Have you been able to replicate that in any of your experiments?
No, so I haven't thought about that at all. And so this likely involves very different brain circuits from the ones I talked about. It might involve the cerebellum and the thalamus. So but I don't have anything intelligent to say about this. So.
Anything that help me remember the stuff I learned a long time ago would be just fantastic. I've forgotten so many things. We have another question kind of related to memory. If the hippocampus is responsible for encoding, storing, and retrieving memories, if you alter the synaptic connections of the hippocampus, can you theoretically make false or synthetic memories?
So we are able to do that. Well, not in my lab, but there's labs who study this. This is a very actively researched question. And yes, so it's in the last few years, people show that it's possible to write artificial memories in the hippocampus. And the way we do this, and so let me bring this up. So from the trivia, is by using optogenetics.
Oh, nice.
The question in trivia. So the way they do it is that because of optogenetics, we are able to activate neurons with laser. So these neurons have special channels that were crossed from jellyfish. So there's some jellyfish that's kind of bioluminescent, something like that. So they crossed those genes from a jellyfish into mice. So now we are able to control neurons with a very specific frequency of light, of laser light. So we can activate neurons by shining a laser on them. And so we can, now we have, imagine we have the hippocampus, this kind of blob of neural tissue. Now we select a bunch of neurons and we just stimulate them again and again and again. So when you stimulate them, these neurons become active, and so the synaptic connections between them get strengthened. And so this way you can create artificial memories. So, and they show that these correspond to artificial memories for percepts that the animals were not exposed to. So you can test them with behavioral experiments. So this is an incredible direction of research that I'm super excited about. Yeah. So,
a lot of fun sharing all the different ways that brains can do things when you visit with your colleagues. Kind of going back to your talk about the sequence of things, have you observed or can hypothesize a maximum speed at which the brain can act? And is it different for different animals? Okay, let's see. I haven't thought about that, but let me speculate from the theory I just presented. So if you remember, so there's 2 ways to control the speed of the sequences. One would be by increasing or decreasing the noise that drives this transition. And the other one is by increasing or decreasing the barrier separating these attractors that generate, right? So the barriers can be controlled, for example, by some behavioral state, like by running or by being very attentive or something like that. So this is a natural way to decrease the barrier and accelerate the sequences. And so I can think about using optogenetics, again, about ways to artificially decrease this barrier so that I accelerate the sequences. However, I think there's a maximum speed at which you can accelerate the sequence, and if you increase the perturbation too much, you're just going to destroy this landscape of attractors, basically. Because if you squeeze all the barriers, then there's no potential ones anymore, there's no attractors, and you have destroyed this coherent brain activity. So I kind of believe that there's going to be, this is a trade-off then between speed and reliability or accuracy, if you want. You can't push it too fast. There's going to be a limit where you completely destroy the coherence of the sequences. And this is a great question, actually, that I have to think about. So it would be fun to figure out an experiment in which we can actually find this maximum speed. Okay, so according to this theory that I presented, then there might be many other different ways to address this problem. All right. It looks like those are all of our questions. So, yeah. All right. Well, thank you so much. I hope everyone enjoyed tonight's event. If you would like to watch the video again or share it with your friends, check out the video section on OMSI's Facebook page or your YouTube channel. And don't forget to follow us on Facebook, Instagram, and Twitter for all updates on future events and inspiring content from OMSI. I know I mentioned this at the beginning, but please consider supporting OMSI Science Pubs and making a donation via Facebook donate button, or you can visit omsi.edu/donate. Join us next week on Tuesday, August 25th for a lecture about the most recent efforts to clean up the Hanford nuclear site. Hanford is home to the world's largest environmental cleanup, and work is ongoing to reduce risks at the site. Learn about the extent of contamination at the beginning of cleanup and the progress and challenges that still remain, including the 56 million gallons of high-level waste stored at 144 aging underground storage tanks. Once again, thanks to our partner Cellstream for helping make tonight's event possible, and of course to Dr. Mazzucato for all of his work and sharing it with us tonight. You can get more information at omsi.edu. Thank you to everyone and have a great night.